Coming up on Harvard Chan This Week in Health, fundamental questions.
Really, a lot of the advances in human health overall have been done by measures of public health. In many areas, those are driven by real advances in the basic science.
In this week's episode, an in-depth conversation with two scientists who are working together to run a unique lab that's trying to crack the secrets of diseases ranging from diabetes to dementia. Hello and welcome to Harvard Chan This Week in Health. It's Thursday, January 11th, 2018. I'm Noah Levitt.
And I'm Amy Montemuro.
Amy, when you read or hear about a new medical breakthrough, maybe it's a new drug treatment for a disease, how often do you think about all the work that went into it? The research, the drug trials, the testing, it can take decades.
And at the heart of all that work is basic scientific research. Before a drug can be developed, scientists must identify a target for that drug. And before that, researchers must understand the basic biology of a cell or an organ.
It's this path of discovery that eventually leads to applications that improve health. But it's not an easy path, and there's a fair amount of serendipity along the way.
And that's what we're focusing on today by talking to two scientists who are asking fundamental questions about how our bodies work.
I'm Toby Walther, and I'm a professor of genetics and complex diseases and cell biology, and also HHMI investigator.
Yeah, I'm Bob Farese. I'm a professor of genetics and complex diseases here at the School of Public Health and a professor of cell biology at the medical school.
For the past 3+ years, Tobias Walther and Bob Farese have worked together to run a lab based here at the Harvard Chan School and the Harvard Medical School. Their work largely focuses on understanding how our cells store and then synthesize fat, a process known as lipid metabolism. And in recognition of the lab's work in 2015, Walther was named a Howard Hughes Medical Institute investigator. The prestigious 5-year appointment provides each scientist with his or her full salary, benefits, and a research budget.
The fact that Walther and Farese are joint leaders of a lab is something that's almost unheard of. And later on in our interview, we'll be talking about the benefits and challenges of working together to run a lab with nearly 24 members. But what's also unique about Walther and Farese is their approach to science.
And you'll hear throughout our conversation, Walther and Farese have a passion for asking big questions about our bodies. And they really want to dig deep to understand what's happening inside our cells.
Their research specifically focuses on lipids, which are fatty acids and oils that store energy and provide materials needed to build and maintain our cell membranes.
And Walther and Farese have a particular interest in something called lipid droplets, the tiny structures in cells that store fat. These droplets play an important role in the ways our bodies function.
One way these droplets work is by storing lipids in adipocyte, or fat cells, for later conversion into energy. This process also protects cells throughout the body from the harmful effects of too much dietary fat.
This system is beneficial to our body during periods of hunger, when food and energy isn't available. But it can break down when people gain and retain excess weight. Farese and Walther are working to understand what proteins or genes might influence these processes. And so far, they've identified more than 200 genes that are critical to energy storage and utilization in our bodies.
If we can understand how they work, then we can understand what goes wrong when they don't work properly and how that gives rise to disease. At the heart of things, we're trying to understand fundamental principles about how life works.
The potential applications of this research are vast, with implications for the treatment of diseases such as obesity, diabetes, and fatty liver disease.
And an emerging focus for the lab is understanding the cell-based processes that underlie specific neurodegenerative diseases, such as dementia, ALS, and Parkinson's disease.
And this is a key theme of Farese and Walther's work. If you can understand how our bodies function at the basic cellular level, it can open the door to new ways of thinking about and treating diseases. They don't want to just scratch the surface of an issue. They want to dig deep and understand root mechanisms.
It's a partnership that's been evolving and strengthening for more than a decade.
We met in 2004 or 5, something in that region.
Early January 2005.
All right, there you go.
You don't remember?
No. Well, we met. I was sort of a postdoc almost on my way out in Peter Walther's lab at UCSF, and Bob was a investigator at the Gladstone Institute, which is part of UCSF, but was also across the street. And he had a more medical background to this point and done a lot of physiology, but had a desire to become better exposed to sort of basic cell biology. That way met Peter and came to our lab, and we met that way. Started a project together and then been working together ever since, essentially.
I had been running a lab for about 10 plus years at that point and had been involved in identifying some of the enzymes that are in a specific part of the cell called the endoplasmic reticulum that make fats. And I got curious about, okay, if they make fats, then how do you store these fats in a cell? How does a cell store these fats? And it was known that they stored them in this part of the cell called oil droplets or lipid droplets. And there was just very little information in any textbook or most of the literature on how these processes occurred. And so, I realized that my training had been in medicine and then physiology and disease mechanisms, but not really in fundamental cell biology, and that to follow that I would have to get better trained in that. So I spoke with Peter Walther, who was an eminent cell biologist at UCSF. He welcomed me into the lab for a sabbatical year, and literally there was an empty desk in the lab, and I sat down next to Toby. And Toby, as he said, was a senior postdoctoral research fellow at that point. And he said, what are you interested in? What are you working on? And I explained it to him, and he said, that's pretty cool. I'll help you with that. And I was like, really? You'll help me? And, because it's not often that somebody who's a postdoc is going to take time out to help some dumb sabbatical professor who doesn't know what he's doing. And, but Toby had both the confidence and the genuine interest in the problem, to do that. And it just, we started working together. It was really fun. And we had a very profitable year where we did a large-scale screen that sort of helped open up the field a bit to molecules involved in this part of biology. And then, in addition, Toby eventually crossed paths with someone who worked in my lab who he's now married to. So.
Farese and Walther say one thing that sets their lab apart is the unique combination of skills they bring to the table.
And at the time they met, their partnership was really novel. Most scientists who were studying fats didn't spend time diving into the inner workings of a cell.
For Farese and Walther, it was an opportunity and a fairly unexplored area. But for that reason, it was also critically important to understand the fundamental biology at play.
And in this approach, Walther and Farese are a perfect match. They relish the opportunity to work together to unravel a problem. But as Walther explains, you can't do that until you understand the basic principles.
There's only 3 types of explanations. Circular ones, endless regress, or causality. And the only explanation type that has been successful in allowing us to modify outcomes is causality as a type of explanation. And so we strongly believe that you understand fundamental principles and how things happen, what leads to what, and that type of causality will lay the foundation for all attempts to understand physiology, pathology, and allow us to develop therapeutic avenues. And that has, I think, not only in our work, but if you look around, I think that generally pans out. Sometimes there's serendipity and discovery, but on the long run, most of the things that work, one can understand from a fundamental perspective.
So we like to ask fundamental questions, like really how do things work from a fundamental principle standpoint, and try to understand mechanistically in as much detail as we can how something happens. And then I think both of us are very in tune to how does that scale. If we understand it at a molecular level, for example, if we understand how a certain protein is involved in storing fat in cells, how does that play out in processes that involve fat in an organism? How is fat absorbed? How is it stored? How is it moved around the body? That kind of thing. And then how does it go wrong? In cases where people have way too much fat, for example, what goes wrong? So we have an eye towards all of that. But I think our orientation, which again is like how we see the world scientifically and partly why we do this together, is like we're trying to understand things at a fundamental level as our central mission, and with us and other people translating these into the larger organismal background or a disease background.
But Walther and Farese admit there are challenges to this approach. For example, they sometimes have to corral their natural curiosity because they risk going off on an unfocused or unproductive tangent.
And that challenge, they say, is magnifying when managing a lab of 20 plus people, all of whom are smart and bring their own ideas to the table.
I find that one of the biggest challenges is to find the right balance between nearsighted focus on detail of an experiment where you can do an experiment essentially every day, and a vision that means the project, we want to have a project that after 5 years solves a fundamental problem, which might have tens to hundreds of experiments, and help people navigate on a day-to-day level to build those steps that will eventually get them to that final goal. So that, I think, is sort of the challenge, to have a long-term vision, but focusing on a detail today to make that happen. So I think everybody struggles with that. That's certainly not unique to our lab.
When it comes to focus and prioritizing, Walther and Farese say that's when they really see the value of running a lab together.
They can bounce theories off each other, help each other interpret data, and importantly, weed through and filter out the bad ideas.
There's also the day-to-day challenges of dealing with large-scale projects, such as Walther alluded to. In their lab, projects can range from 2 years on the short end to up to 4 to 5 years on the long end. And Farese say he and Walther have an important responsibility to the other scientists in their lab.
Yeah, the kind of work that we do, a lot of bench science work, it's incredibly hard work day to day. And part of our job is to see where we think the end outcome is going and help to keep things moving in that direction. It's also to be a cheerleader sometimes. There's a lot that happens over several years of someone's life. We have a lot of young people who are literally maturing in front of our eyes as young investigators and young people in general. And so it's a really interesting environment. It's fascinating from a mentoring standpoint, and it's also, I think, it gives us a lot of satisfaction too to see people embark on these big projects, bring them to fruition, be really proud, because generally people are very excited about something they worked quite hard for at the end of the day. And if we put our seal of approval on it and publish it, we're all very, very proud of these contributions.
And this management and research style has proven fruitful for Walther and Farese. As we mentioned earlier in the episode, they have identified hundreds of genes that affect how cells store lipids.
And now that they've deepened our understanding of that mechanism, they're taking a closer look at what happens when a cell's capacity to store fat is overwhelmed.
And this, in turn, triggers a bunch of pathways in the cell to try to deal with those lipids and restore homeostasis. However, if that becomes overwhelmed, then it often can trigger cell death. It can trigger inflammatory pathways, and so forth. So an example that's very common right now is that accompanying obesity, many people store too much fat in their livers. So for some people that goes okay, they just have fat in their livers. But for another portion of those people, this triggers consequences to the cell that are detrimental, either cell death or inflammatory pathways. The inflammatory pathways in particular stimulate a kind of fat-induced hepatitis, and that can trigger cirrhosis of the liver and liver failure. And this is a big current public health problem of liver failure due to too much fat in the liver. So, we're trying to understand those processes.
And understanding this process is critical because in addition to the liver failure that Farese mentioned, excess fat can interfere with the body's ability to process glucose, resulting in type 2 diabetes and other cellular malfunctions that can often lead to heart and blood vessel damage.
We've actually created a visualization of what this looks like when cells get overwhelmed by fat, and you can view that video on our website, hsph.me/thisweekinhealth.
As Farese and Walther work to understand these processes, they're filling in critical gaps in our knowledge about how our bodies and cells work.
If you come to one of our lectures on this topic, what you would see is that I think when we started this, basically we kind of know it starts like there's two things on the slide. It starts here and ends here, and that's about all we know. And then we hope that what we've helped to do, in the past decade or so, is begin to put a lot of molecules on these pathways and build an understanding of at the end of the day, cells use proteins to carry out as machinery to carry out different processes, and so we begin to understand some of the concepts that are involved. And we still have quite a ways to go with what we study, but we think that we've made some contributions to that.
This path of discovery for Farese and Walther hasn't been a linear one. In fact, they say science rarely is.
Walther says that often as a scientist, you don't know exactly where your research will take you.
There is serendipity and discovery along the way, and often there are things that you do not anticipate as being important that turn out to be really important, and then you have to go down that road. And those are some of the most critical decisions we make collectively in the lab, is like, is this an important thing to follow or not? And it's the hardest part, because you can go in the wrong direction for a while and then you have to turn back.
Farese agrees, saying that as a lab, they generally try to set out on a linear path, but often find themselves needing to change course.
I basically came up into the field working on cholesterol metabolism and studying enzymes that were involved in cholesterol metabolism. And, the way I view it is like we kind of, we're captaining a ship and we set our course towards that lighthouse over there and we get going, but then we have to kind of keep our eyes open because we never know what we'll see on the way. And in that way, in that manner, in studying cholesterol metabolism, we're cruising along and, banging our head against the wall trying to elucidate the function of one gene that we'd cloned and thought it was involved in cholesterol metabolism, and literally, on a lab meeting, seeing a band in a completely different part of the gel that told us that this was not cholesterol metabolism at all, but it was, involved in fat or triglycerides, and having to keep your eyes open to that possibility, because if you don't, you miss it and you move on.
And an important takeaway from our conversation was that scientists need to have this freedom to discover, to take winding paths where serendipity may eventually lead to a breakthrough.
Farese and Walther told us that most major discoveries don't start with an application such as a treatment in mind. Instead, they start with a desire to understand basic biology, and that opens the door to finding a useful application.
But that translation of research, such as a drug target, can't happen until you understand the basic biology of a disease or a process in the body.
It's an incredibly important and difficult part of the research process. Farese illustrated how by discussing the lab's recent work on Alzheimer's.
We also work, on this frontal lobe dementia project where there's a protein called progranulin, and if you're low on that protein, you can get frontal lobe dementia. But 10 years after the discovery of that, we're still trying to understand at the cellular level what this protein does. And we don't yet understand that. If we do, I think we'll have a much better idea of trying to find treatments for deficiency of that protein, and we may even have a much better idea of finding treatments for a whole host of neurodegenerative diseases because we understand the basic principle that's involved.
I also think, to some degree, if you look at the most common indications and most common diseases, a lot of the obvious simple things have been done. And I think to some degree, to really push new frontiers, you do need a better understanding, because there are certain limitations of what you can try more or less randomly. And so for that to happen, I think there needs to be more discovery, and you can make a great argument. For instance, in our space, one of the more prominent new therapeutic targets is PCSK9, and that's a protein that is now used for lowering potentially cardiovascular outcomes. That was a long process to come to that, and it started from a number of different observations, all made in basic science, all have to do with regulation of lipid metabolism on the one side, with mutations in families that were analyzed. So different branches of science had to come together and sort of provide this evidence, and I don't think you can do that with just saying like, all right, let's this one approach is going to give you all the targets.
A key to understanding basic biology is the access to more powerful tools than ever before, especially when it comes to imaging technology.
Farese and Walther say we're only just beginning to tap the future potential of this technology.
For me, this is the early 20th century in physics, is what we are going through right now in some way. For most of the last 20, 30 years, honestly, I mean, there was no tools to study some of the most important problems. And then, people started to study, use genetics, has come a long way. But how technology has accelerated this is absolutely phenomenal. And I think there's really at least 3 or 4 revolutions. I mean, now one of them is that you can analyze mammalian genomes. It started with mouse knockouts, but has now become much more routine, and we can do a lot more things. We have analytics that allows us to really understand the composition of biological systems in every dimension, from the DNA to the proteins to lipids and sugars. And we have imaging technologies that allows us to visualize this at at all kinds of levels, essentially anywhere from the subcellular level, looking at atomic structures, all the way to what happens in an intact organism with intravitreal microscopy, PET scans, and so forth. So I think this is really unbelievable. And I really feel that only in the last 5 or so years this has come together where we can do meaningful things, for instance, in human biology. It was really not there before.
Your example there, the ability to knock out a gene in a mouse, that's when I started my career, basically, was that technology, learning that technology and helping to implement it. And, now that was a lot of work, years of work to knock out one gene in a mouse. Now the ability to knock out a gene in a cell is, becoming rapidly rather routine in laboratories. It's accessible, so people can study gene function in a human cell, for example, very, very much easier. And I think, just science, of course, moves along, and it's not like a lot of the questions have been around a long time, but it just depends on the technologies that allows us to investigate, ask questions, analyze the data, see things we haven't seen before.
And at the heart of that process of investigation is what we've talked about a lot in this podcast, asking fundamental questions.
But a key for Walther and Farese is also being able to ask those questions, get answers, and then scale their discoveries.
The goal is to first focus on universal questions at the core of the issue they're working on, and from there they can branch out and test.
So what does this look like in practice? Farese shared the example of their work on sphingolipids, which are found in cell membranes, particularly nerve cells and brain tissues.
Very little is known about how these sphingolipid concentrations are regulated and so forth. And, Toby, just from a very basic standpoint, recognized that was kind of a fundamental problem that we knew very little about and got interested. And so he and folks in his lab at the time did some, very basic studies of just like, let's take genetics in yeast, let's knock out every gene in yeast and see what affects this. And basically, to make somewhat of a long story short, found some genes in yeast that were important in this sphingolipid metabolism, and it turned out they were related to disease genes that cause like ALS-like problems in humans. And now we're in the process of trying to translate that into mouse and or human studies, but we're actually doing a little mini clinical trial in a mouse study where knowledge we got from this new connection between sphingolipids and an ALS-like disease, we're testing that in a mouse model. And that's very exciting and basically shows the principle of just starting with a fundamental observation, making some observations, and then recognizing the potential to understand something that might be useful, for example, in human disease.
And what makes this all possible, says Farese, is the creative synergy that he has with his partner.
I think it's hard to find the right person to do that with. You really have to agree on a lot of values, and you have to get to know each other pretty well, and really figure out ways that you support each other through these processes. It's just like anything in life, is there's joys and struggles, and it's hard, and but I think also part of it is it's really fun. Basically, we have adjoining offices with pocket doors, and we talk about everything. We talk about science, we talk about management issues, we talk about school politics, whatever it is. We talk about it, and I think we both learn a ton from each other that way. It prompts our thinking. We end up kind of coming out with pretty similar thoughts on many things based on this, but that's natural because of that. But it just makes it a lot of fun. And as Toby mentioned, I think we both ran labs individually for quite some time before joining the labs, and everything has its trade-offs, but I think it can be a lonely experience running a lab because when you don't have somebody to talk to for all the things that you're struggling with. And I think it's been it's a wonderful thing to be able to share whatever your both your joys and your struggles with someone.
We thought that was actually the end of our interview, but Walther made a really important and powerful point.
We've talked a lot in this episode about the importance of basic scientific research, of asking fundamental questions. And Walther says it's important to consider that work in the larger context of public health, how the discoveries being made in a lab today can be used to address some of the world's major challenges in the future.
Really, a lot of the advances in human health overall have been done by measures of public health. In many areas, those are driven by real advances in the basic science. What we're facing now as a planet, one major challenge is non-communicable diseases and chronic diseases. And those are really taking over the planet in every region of the world as major problems. And as a consequence, you have failing healthcare system. Lots of policy work needs to go into this. But also it really highlights the need to understand better on the level of epidemiology of what are the contributing factors, but then on the level of science, basic science, what are the causal chains that actually lead to all these problems. And really, when you think that through, at the end, one crucial component to this is understanding fundamentally how the basic biology works that then has all these implications. And one thing that's really fabulous here is that we're sort of the only place in the world where that is brought together under one roof, and where we actually have both people like Bob and myself and the people in our lab who think about, the structure of a protein and how this really works, all the way to people who think about, how, for instance, effects that could be mediated by saturated fatty acids and how that translates to a population and what should be policy recommendations based on that. And bringing that together is a really powerful thing, I think.
That was our conversation with Tobias Walther and Robert Farese.
If you're interested in learning more about their work and their lab, we'll have much more information on our website, including that really cool visualization of cells and lipids. And that's at hsph.me/thisweekinhealth.
Coming up in next week's episode, new research shows that many large chain restaurants are dropping high-calorie items from their menus. We'll talk with the author of the new study about what these findings mean for America's fight against diabetes and obesity.
In the meantime, you can always listen to this podcast on iTunes, SoundCloud, or Stitcher.